Hello, everyone, and welcome to today's live poster presentation, In Vitro Research Method for Screening Inhibitors of Protein Translation, presented by Dr. Krishna Vattam, Senior R&D Scientist, Protein and Cell Analysis, Thermo Scientific. I'm Julie Simmeroff, and I will be your moderator for this educational webcast, presented by LabRoots and sponsored by Thermo Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the poster in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Please join me in welcoming Dr. Krishna Vattam. I will now turn the presentation over to him.
Hello, everyone. Thank you for joining me for today's poster presentation. Today, I'll be talking about a very simple in vitro cell-free expression method that can be used for screening of inhibitors of protein synthesis. Inhibition of protein synthesis can be used as a mechanism to block uncontrolled cell proliferation, and this feature is gaining popularity as a method for treating cancer. As you know, all eukaryotic mRNAs contain a cap structure at the 5' end. Using a dual luciferase mRNA construct containing 5' cap, turboluciferase, and an IRIS red firefly luciferase, and in combination with our cell-free expression system called one-step human IVT, one can screen for drugs that inhibit translation of eukaryotic mRNA. Inhibitors that specifically affect the cap mRNA translation generally do not affect IRIS or internal ribosome entry site containing mRNA translation. And this feature can be used to monitor the specificity of the drugs affecting the cap mRNAs, which are in fact present in all the eukaryotic mRNAs. Since IRIS containing mRNA are common among viruses, our assay can also be used to screen for inhibitors of viral mRNA translation. Our dual luciferase assay can be carried out in the same tube or well of a 9 to 6 well microtiter plate and is very rapid, just takes about 60 minutes, and can be also miniaturized to as little as 10 microliters for a high throughput screening format. This is how the overall poster looks like, but I'll be going into each of the individual results to explain in detail. I'm going to move forward into slide 4, where we go directly into the results section, and of course the method section before that as well. This figure shows the assay protocol. Basically, the materials you need for this experiment include mRNA templates, cell-free expression reagents, or the IVT reagents, a 9 to 6 well microtiter plate, luciferase assay reagents, a luminometer, and of course your compound that you want to test if they affect protein synthesis. So the configuration of mRNAs is as follows. One mRNA has a 5' cap structure and turboluciferase as a readout, and the other mRNA has an IRIS structure at the 5' end and a red firefly luciferase as a readout. The cap structure is added while preparing mRNA using an in vitro transcription reaction. We will talk about this step again in the next slide and see which capping mechanisms work better. Now it's easier to make a single master mix of all the IVT components just to reduce the number of components you need to add to the microtiter plate. If you prefer, you can add the mRNA templates to this master mix as well. so it becomes just a one-part mix. I usually add the compound to the microtiter plate and then subsequently add the IVT, now I call the master mix, which contains all the IVT reagents and the mRNAs. So it's basically a two-step addition. The compounds first and then the IVT master mix. This way, it's just, as I said, just two additions. Your compound that you want to treat has to be present in as little as, can be present in as little as a microliter to 2 microliters in a microtiter plate. So you let the reactions incubate for about 60 minutes at 30 C. And I usually take about 2 microliters at the end of the reaction to measure the turboluciferase and the red firefly luciferase. In a typical experiment, if your compound affects the cap-dependent translation, you may see inhibition of turboluciferase, which is a readout here, while the red firefly luciferase activity remains more or less unchanged. The compound affects the activity of both the luciferases, then it might mean that the compounds affect something else in the protein synthesis process. It could be the ribosome assembly or some other steps in protein synthesis, including initiation and elongation. As I mentioned before, eukaryotic mRNAs have a cap structure called the M7G, or the 7-methyl-guanosine, in their 5' ends, and this is required for efficient translation. One can in vitro add a cap structure to mRNA while carrying out the transcription step and while generating the mRNA templates. In order to determine the best mRNA capping protocol, we compared 3 different systems to find out which method is the best in terms of getting maximal expression of the reporter gene. In our case, it's the turboluciferase. One has to keep in mind that the cap structure can be incorporated in either orientation during the transcription reaction. To specifically address this issue, we have generated what is called an anti-reverse cap analog, or called the ARCA, in which the 3' hydroxyl is eliminated and the 7-methyl-guanosine or the cap structure, and it is replaced with an alkoxy group. This facilitates RNA polymerase to initiate transcription with the remaining hydroxyl and the guanosine, and thus the synthesized transcripts or the mRNA or the cap mRNAs are exclusively with the correct orientation of the cap structure. As you can see in the results, the ARCA-based method compared to 2 other methods from our competitors is working the best in terms of giving the maximal expression of luciferase. So in all our subsequent protocols, we in fact use the ARCA-based capping method to synthesize our turboluciferase mRNAs. Ideally, to test new inhibitors of protein synthesis, the effects of both cap-dependent and cap-independent translation should be simultaneously evaluated. To optimize our expression system for this dual screening method, we evaluated 3 different combinations of mRNA additions. As seen in this figure, although maximal expression is obtained when only a capped message was added to the system, as you can see in the A, which is the leftmost of the graph. And there is a moderate reduction of translation of mRNAs when both the capped and the uncapped or the iris containing messages were added to the system. In this section, in the middle part of the graph, you'll see that both the messages were added as separate components, but at the same time. But in section C of the graph, you see that the message was added as a bihistonic mRNA, where the cap structure was present at the 5' end of the message, and then in the middle you see an iris structure followed by the red firefly luciferase. This is a matter of convenience too, where the additions would become just 1 mRNA instead of adding 2 mRNAs. Now, we come to the actual experiment where we begin to test the effects of various compounds or inhibitors on the expression of mRNAs. As you know, protein synthesis is broadly divided into initiation, elongation, and termination, and the inhibitors can affect any of these steps. We choose inhibitors such as M7-GDP, which is a non-hydrolyzable cap analog that would specifically affect the mRNA containing the cap structure and therefore the expression of turboluciferase. We also included inhibitors such as pyromycin and cyclohexamide in this experiment that are known to affect other steps in translation, namely the elongation step, where you would see a reduction of both the turboluciferase as well as the red firefly luciferase, which means these compounds affect the translation in general and not specifically at the cap structure. In addition, we also evaluated the effects of these compounds using two different cell-free systems. The top one is pertaining to the HeLa cell-free system, and the bottom one is pertaining to the CHO, or the Chinese hamster ovary cell-free system. As seen in this figure, both the cell-free systems give almost identical results, where cap-specific structures inhibit turboluciferase, and inhibitors which affect both the elongation step, such as cyclohexamide and pyromycin, affect this translation of both the messages, the capped and the uncapped messages, as you can see here. As a control, we use ribavirin, a viral replication inhibitor, which affected neither of these templates. Taken together, these results indicate that you can choose either of the mammalian cell-free systems, HeLa-based system or the CHO-based system, to screen for protein synthesis inhibitors. Furthermore, these experiments were carried out in 10 microliter reactions. You could even go down to 5 microliters if you choose to. This suggests that the entire experimental setup can be miniaturized to suit a high-throughput screening format, and this can be done using just on a tabletop experiment instead of using an entire cell culture experiment to get these results. Finally, using our in vitro protein expression system, we wanted to determine if one could obtain IC50 values of the inhibitor compounds that we assessed in the previous experiment. As you can see, varying concentrations of inhibitors were added and IC50 values were obtained for both M7-GDP as well as for pyromycin on the right side. The values for M7-GDP were about 10 micromolar, and the IC50 values for pyromycin was about 87 micromolar when using turboluciferase as a readout, which is a capped message, and about 27 micromolar when using red firefly luciferase, which is dependent on iris. It's a non-capped message. In conclusions, the one-step IVT system is capable of screening protein synthesis inhibitors in a miniaturized, fast, high-throughput screening format. You can simultaneously monitor the cap-dependent and cap-independent translation using the turboluciferase as a readout for the capped messages and red firefly luciferase as a readout for uncapped or iris-dependent messages. The dual luciferase as a system, which is measuring both the turboluciferase and red firefly luciferase, provides a very unique advantage, or rather advantages. Here, identification of inhibitors specific for cap structure regulation can be carried out by capped turboluciferase mRNA expression, while simultaneously evaluating its effect on general translation by monitoring the effect of cap-independent translation. Iris structure is very specific to viruses and can be used as a target for screening viral inhibitors as well. Lastly, the flexibility of the system affords one the ability to replace the EMCV iris with the other irises, for example, from cricket paralysis virus or hepatitis C virus. These irises are known to promote translation in a cap-independent fashion. So we can take the advantage of this fact and, in fact, use this as a method to screen for inhibitors that specifically bind to the irises. This concludes my presentation, and I'll be happy to take any questions. Thank you for your attention.
Thank you, Dr. Vatum, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. The first question is, can you tell me more about the cell-free system that is discussed in the poster?
Sure. So this cell-free system is a very simple system. This is made up of, we have two cell-free systems, mammalian cell-free systems, or it's also called as IVT system, by the way. So this system is made out of a couple of components, a cell extract, in which case it's a HeLa cell extract or a CHO cell extract. And in addition, we provide all the components in one mix called the reaction mix that contains the energy generating system, nucleotides, and other factors required for efficient translation. All you need to do is mix the lysate and the reaction mix and add the DNA or mRNA of your interest to this mix. And voila, you'll get expression of proteins in as little as 60 minutes.
Thank you. The next question is, can this cell-free system be used to express high molecular weight proteins?
Yes, of course. One of the fundamental aspects of cell-free expression system is expression of proteins itself. And what we presented today is an application of the cell-free system that can be used for screening viral or protein synthesis inhibitors. So coming back to the question of advantages of cell-free systems, yes, you can express proteins, and you can express using the HeLa or the CHO cell-free system, you can express proteins as large as, I think, 270 kilodaltons is what we tested. But the system, we didn't have DNAs that code for even larger size proteins, but I'm sure you can do that.
Thank you. The next question is, how does this IVT system compare to the other cell-free systems that are available?
So there are several systems available in the market. We call our eukaryotic cell-free systems as IVT systems, in short for in vitro transcription translation systems. So ours is a mammalian system. As opposed to a bacterial system, our system is capable of producing, incorporating post-translation modifications into proteins. Bacterial systems do not. And in addition, our system is a mammalian system, which is the most popular and most directly applicable system to researchers who carry out cell culture work propagating mammalian cells. So I would say this has advantages over other systems which include plant-based systems, where you're probably expressing a mammalian protein in the plant system, which is not ideal, but using our system, you can express mammalian proteins in a mammalian system. That is our advantage.
Great, thank you. You mentioned the system could be used in an HTS format. What is the minimum reaction volume required for this assay?
Yes, that's a good question. So as I mentioned in my slide deck, we went up to, I think, 10 microliters minimum volume, but I'm sure you can go down to 5 microliters volume as well. The reason is the way you set up the reaction. You can essentially set up the entire reaction as one master mix. This one master mix would contain lysate, which is essentially the guts of HeLa cells or the CHO cells, and which contains all the ribosomes, all the factors in it. And you can mix this lysate with the reaction mix components, and you can make it basically one mix. And in fact, if you're screening compounds or screening inhibitors of protein synthesis, you can make a mix of lysate, reaction mix, and the mRNA as one mix. And you can basically pipette that straight into the microtiter plate, which already has compounds placed in them. So the ability to make one part mix is what facilitates miniaturizing the reaction to as little as 5 microliters.
Thank you. If there are no more questions, I'd like to once again thank Dr. Vatum for his presentation. I would also like to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.